Welcome to our clinical microbiology class. Today we will begin with the most essential chapter in every microbiology laboratory, that is microscopy. So what is microscopy? Microscopy refers to the technique in which the use of a specialized instrument that is called microscope for the visualization of the objects that cannot be seen with the naked eyes. So microscopy includes light microscopy, electron microscopy and scanning probe microscopy. Light microscopy, in which light is used for visualization of the microorganisms. Electron microscopy, in which electron beam is used for visualization of microorganisms. And scanning probe microscopy, in which probe is used for scanning of the organisms. Light microscopy mainly includes bright field microscopy, dark field microscopy, phase contrast microscopy and fluorescence microscopy. Electron microscopy includes two types. First is the scanning electron microscopy, second is the transmission electron microscopy. Then scanning probe microscopy, here the probe is used for scanning of the microorganisms. Example is atomic force microscopy. Various types of microscope that are used in diagnostic microbiology. First, bright field or light microscope. Second, dark field microscope. Third, phase contrast microscope, fluorescence microscope, electron microscope and atomic force microscope. What are the different properties of microscope? First, it requires good resolution, good contrast, good magnification. So what do you mean by good resolution? So resolution means two closely placed objects appear as two separate entities. It depends upon the refractive index of the medium. Oil has a higher refractive index than air. Hence, use of oil enhances the resolution power of microscope. So here in the picture, on the left side, it shows poor resolution, and in the right side, two closely placed points are more clearly defined. Good contrast, this can be improved by staining the specimen. When the stains bind to the cell, the contrast is increased. Third is the good magnification. This is achieved by use of convex lens. So total magnification can be calculated as eyepiece lens into objective lens. Suppose thus we can calculate the total magnification of low power objective lens, in which the ocular lens is 10x and low power objective lens is 10x. So total magnification will be 100x. So likewise, the total magnification of the objective lens can be calculated. So magnification making image appear more larger, and resolution mainly clarifies the image. So unaided human eye, the resolution power is 200 micrometer, light microscope 0.2 micrometer and electron microscope 0.5 nanometer. So what are the various parts of the microscope? It starts with from below, light source, then condenser, iris diaphragm, stage, then three types of objective lens. One is the low power objective lens, high power objective lens and oil immersion lens. Then ends with the ocular lens. So what is the working principle? So light mainly pass through the iris diaphragm, then to the condenser lens, through the specimen to the objective lens, and both the reflected and unreflected light mainly pass through the objective lens and finally passes through the ocular lens, in which we can see the magnified image. So bright field microscope mainly forms a dark image against a brighter background. So uses examination of gram-stained bacteria, Zn stain smears for the acid-fast bacteria, observation of stained blood film, fungi, parasites and tissue sections. It is mainly used as the routine diagnostic microscopic procedure. So what is dark field microscope? That is bright image and dark background. So what the working principle here? A dark field condenser is used to the central opaque area and peripheral annular hollow area. Here the light passes through the dark condenser in which the central opaque area which does not allow any light to pass through it, and the light mainly passes through the annular hollow area which is present in the peripheral part. Then the light falls on the specimen, and from the specimen the unreflected light does not pass through the objective lens. Only the light reflected by the specimen, that is reflected light, is mainly captured by the objective lens, and finally it falls on the ocular lens and we can see the magnified image of the object. What is the applications? Used to identify the living unstained cells and thin bacteria like spirochetes which cannot be visualized by light microscopy. So what is phase contrast microscope? Here contrast is enhanced, visualizes the unstained living cells by creating difference in contrast between cells and water. What is the working principle? Here condenser is similar to dark field microscope, that is cone of light passes through a cell. Some light rays are bent due to variation in density and refractive index within the specimen and are retarded by 1/4 of a wavelength. So undeviated light rays strike a phase ring. Deviated rays miss the ring and pass through the rest of the plate. Background is bright while the unstained object appears dark. Phase contrast microscope. So here in the picture, the undeviated ray mainly pass through the phase ring, and the deviated rays which mainly pass through the specimen, they mainly deviated by specimen is 1/4 wavelength of out of the phase, and they mainly misses the ring and passes through the other parts of the phase plate, and deviated ray is half wavelength out of the phase. So both the deviated and undeviated rays cancel each other out. The light rays go through condenser, then specimen, then phase ring, objective lens, and finally on the ocular lens. So here in the picture, the dark ground microscope picture demonstrating spirally coiled bacteria, that is spirochete, and in the B, phase contrast microscopic picture demonstrating Naegleria fowleri trophozoites. What is the application of the phase contrast microscope? For studying bacterial motility, determining the shape of living cells, detecting microbial internal cellular components. If you enjoyed the session, don't forget to like, share and subscribe. See you all in the next microscopic chapter. Thank you.